In this presentation, I'm going to talk about the reproduction of bacteria through the process of binary fission. Before we start to look at bacterial reproduction, we need to be familiar with the structure of the bacterial cell. You can be asked to label this diagram in the exam, but not to draw it. To start off, the very outer layer of the bacterial cell is called the capsule. The capsule provides the bacteria with a layer of protection during reproduction and growth. Inside the capsule then you have the cell wall. This is a rigid and tough section of the bacterial cell that gives it structure and shape. Inside of the cell wall then you have the cell membrane. The cell membrane holds together the cytoplasm that is inside the bacterial cell. The cytoplasm then contains ribosomes and strands of DNA. Note that in a bacterial cell, the DNA is just in a strand and not in a nucleus. The last part of the bacterial cell we need to look at then is the flagella. The flagella is like the tail that allows the bacterial cell to move during growth and reproduction. Bacteria reproduces through a process called binary fission. I've included the 4 steps of bacterial reproduction here, both in written and diagram form. In the exam, you can absolutely use a diagram to aid your discussion, but definitely you would need to include some words or description, or else it's just a pretty picture that's not going to get you any marks. So starting off, step 1, you have the bacterial cell that obviously has favourable conditions. So the correct temperature, enough food, oxygen or not oxygen, depending on what the bacterial cell would like. When it has enough of these conditions, what the bacterial cell will do is it would elongate. Elongate basically means it gets longer, and the nuclear or DNA material that's inside the bacterial cell is going to split into 2 parts. One part goes to one part of the new bacterial cell, and the other part stays with the older part of the bacterial cell. Once it elongates and now has 2 sets of DNA in both the old cell and the new cell, a cell wall and a membrane will form between the bacterial cells to divide them. Bacterial cells divide very quickly. They usually double every 20 minutes. This is until they run out of the favourable conditions like food or oxygen, or the temperature becomes not correct for them. During this stage then, the bacterial cell may form endospores, which we will look at later on. In this video, you can see the bacterial cells elongate and divide, and you can also see how rapidly it occurs to multiply the amount of bacteria. In this diagram, we're going to look at the growth phase of bacteria. Bacteria grow in 4 different phases. The first stage is called the lag phase. During this process, the bacterial cells are adapting and getting used to their new environment. So the amount of new bacteria reproduced is relatively low. The next stage is the log phase. During the log phase, the bacterial cells are multiplying rapidly and the numbers increase dramatically. The next stage is the stationary phase. During this stage, the same amount of bacteria are reproduced that are dying, so there's no increase in numbers. What will kill a majority of the bacteria is the toxins that they are producing during reproduction. These toxins, although new bacteria are being reproduced, are still killing the old ones, which means there's a steady amount of bacteria. It's not increasing or it's not decreasing. The final stage then is the decline phase. The number of bacteria dying is now greater than the number that is being reproduced. A small number of bacteria will survive this stage by becoming spores. The rest will die away, and the usual reason for the decline phase can be a change in conditions. Maybe it could be a change of temperature, a lack of food, or a change in the oxygen conditions. As mentioned in the previous slide, during the decline phase, when conditions for the bacteria are becoming unfavourable, most bacteria will die, but some will survive through the production of what's called endospores. Endospore is basically a tough, protective layer the bacteria will form around its cell of DNA. Its strand of DNA can then survive inside the cell for months or even years until conditions become favourable again. When the endospore finds more favourable conditions, the tough wall breaks down and the DNA duplicates and creates a new bacterial cell. This bacterial cell will then continue to reproduce again through binary fission, which will make the bacteria multiply rapidly. Endospores are very difficult to destroy if they have been formed. For moist heat, you need to heat the food to 121 degrees for 15 minutes, so that's boiling or steaming, and then dry heat, which is like roasting or baking in the oven, it needs 150 degrees for 1 hour. Ideally, these endospore forming bacteria are not to be present in food because they are so difficult to destroy. During periods of rapid growth, bacteria produce toxins. These bacteria are usually pathogenic bacteria, which are the ones that can cause diseases in animals and plants. These toxins that are produced by the bacteria during this time are poisonous to us humans, and that's why they create food poisoning. The bacterial toxins can be divided into two groups: endotoxins and exotoxins. Endotoxins are ones that are made inside the bacterial cell as the bacteria grows and reproduces. When the bacterial cell dies, these toxins are released from the bacterial cell. The good thing about these ones is they're easy to destroy by normal cooking temperatures, but they can cause what's called infectious food poisoning. This is food poisoning that is contagious to those around you. The toxin only infects the area of infection also, which means generally you'll only get symptoms in your digestive system. Examples of endotoxin producing bacteria are Salmonella, Listeria, and E. coli. It's important to know one example from the three of those. Exotoxins then are toxins secreted or released from the bacteria as they grow. They may be produced before the food is eaten or after. These types of toxins are very difficult to destroy and require cooking for at least 30 minutes at a high temperature. These toxins also produce what's called toxic food poisoning, which means it's not contagious but has more serious symptoms. The reason why this type of food poisoning can be more serious is because the toxins not only affect the area of reproduction, which in most cases is the digestive system, but can also affect other areas of the body. Examples of bacteria that form exotoxins are Staphylococcus aureus and Clostridium botulinum. I would know one example of these.